Good afternoon. My name is Salam Saloom. I'm a postdoctoral researcher at Qatar Biomedical Research Institute, or QBRI. It's my pleasure to welcome you all to a new webinar for early career researchers. For those attending for the first time, this webinar series provides a platform of valuable tips, lessons, and PhD career stories to inspire young scientists. If you missed our previous webinars, you could watch a recording on the HBKU YouTube channel. Today's webinar is structured to have a presentation from our guest speaker for about 30 minutes, then an interactive discussion with our speaker, and the last 15 minutes will be for questions from the audience. Just a reminder of housekeeping roles. This webinar is interactive, so please send your questions using all panelist options in the Q&A tab, whether you are using a laptop or a mobile device, as you can see in the slide. So today's topic is about In Pursuit of Knowledge, Lessons Learned from Life in Science. What our speaker will discuss today is precious information for researchers in different stages of their careers, particularly the origin of good ideas, the importance of leadership, teamwork, and collaboration. The discussion will also reflect contemporary concerns related to reproducibility crisis, predatory journals, and reimagining research. So I'm very pleased to welcome our speaker who joined us from QBRI. Dr. Paul gained his BSc in chemistry at the University of Manchester and a PhD in biochemistry at the University of Oxford. Before joining QBRI in 2018 as the Research Director of Diabetes Research Center, Dr. Paul has held lecturer and professor positions in several prestigious universities in the UK, including Aston University in Birmingham, the University of Essex, and the University of Warwick. He has 529 publications, 14 patents, more than 33,000 citations, and an Edge Index of 96. Dr. Paul is a leading international diabetes researcher influencing many in his field, particularly studies of reactive metabolite, Methylglyoxal, in developing chronic vascular complications of diabetes, in which he is considered a founding investigator. Dr. Paul, welcome, and thank you for your time today. We look forward to learn about your long scientific and professional career. I'll pass you the floor now. The control is yours.
Assalamu alaikum, everyone. It's a great pleasure to give the presentation today. I would like to thank Dr. Salam and Dr. Adviti for inviting me to present to the Early Career Researchers Symposia series. So I would like to give a talk about In Pursuit of Knowledge, Lessons Learned from a Life in Science. I am Paul Fornelli, Research Director of Diabetes Research Center here at QBRI. So as the Prophet, peace be upon him, said, seeking knowledge is an obligation upon every Muslim, both men and women. I will briefly describe how I was inspired to pursue a scientific career and the following aspects: inspirational teachers and researchers, scientific literature and dedication, teamwork and collaboration, and contemporary concerns, the reproducibility crisis, predatory journals, and to reimagine research. My interest in science started in a small rural village in Lincolnshire, England, and you can see the location of Lincolnshire on the map on the top right-hand side of the screen. It's an area known for agriculture and crop production, and the centre picture shows a typical picture of a semi-wooded valley in North Lincolnshire, which to me looks beautiful, and indeed I hope it looks beautiful to you too. I grew up on a small family farm managed and worked by my father and my uncle. And on the left-hand side at the bottom there, you can see a typical scene of ploughing in the fields, and on the right-hand side, harvesting in the wheat fields in summer. But my interest in science began in the school in the centre bottom there, King Edward VI Grammar School, Spilsby, Lincolnshire. And at that time, we had a chemistry teacher who was unusually well qualified for a teacher in chemistry at that time. He had a bachelor's degree in chemistry and a master's degree in chemistry, and was a real inspirational teacher. He said, besides giving us tasks to do in this main syllabus, if you get this done, I would like you to look at this more advanced problem, which has real life applications. He also was a polymath. He could teach mathematics, he could teach English literature, the arts. And this knowledge without limitations really was very attractive to me. And I'm grateful to him inspiring me and guiding me into a career in sciences, initially chemistry. And this is why I have strong support for Qatar Foundation and Her Excellency Sheikh Hind's efforts to encourage teachers in the nurturing of talents in children in schools and increasingly online. The first studies I did was a BSc in chemistry in the University of Manchester, and I worked hard and got a first-class degree. The final year I did an undergraduate research project where my tutor at that time said, Paul, you read about the scientific literature about people pursuing their own research questions. If you want to ask some questions yourself and pursue them, then you really need to do a PhD. And so that launched me on to studies for a PhD. And already at that time, I was reading the scientific literature diligently, corroborating evidence, cross-checking, and studying methodologies, both the strengths and the limitations. I went to Oxford to do my PhD under Professor Alan Hill. And at that time, studies of reactive oxygen species, ROS, was becoming very popular in biomedical research. And there was an emerging technique called spin trapping, where we use a nitrone called DMPO, which traps the reactive oxygen species and gives us a report of the formation. And I quickly became an expert and leader in the technique of spin trapping. In fact, in my PhD, I applied this to 5 projects. The first one was free radical involvement in drug-induced hemolysis, where example there I detected by spin trapping phenol radical production in the phenylhydrazine-induced hemolysis. And on the right, you can see a quite complex mechanistic scheme, which has become quite common in my publications. And in those days, of course, I had to do all these schemes by hand. Project 2, I studied free radical involvement in vitamin K-dependent carboxylation of glutamate in blood coagulation proteins. This hypothesis turned out not to be supported by experimental observation. And this was the first lesson that I learned early in my studies, that if the hypothesis is not supported by experiment, it's incorrect. And you need to either develop a new hypothesis or at least modify, refine the existing one. Project 3, we studied free radical involvement in paracetamol overdose toxicity and development of cysteine and methionine antidotes. And in fact, methionine is co-formulated with the paracetamol currently in a so-called safe paracetamol formulation. In Project 4, I studied free radical scavenging by a cysteine-rich protein, metallothionein, and this paper currently has 1,255 citations. It's my most highly cited publication currently. And Project 5, I studied free radical formation by a phagocytic NADPH oxidase enzyme, a professional ROS generator. And this brought me my first international collaboration. Professor Filippo Rossi, you can see in the photo on the bottom left there, was an expert on human neutrophils and based in the Institute of General Pathology in University of Verona, North Italy. And like me, he was a very dedicated researcher and a scholar of the literature, scientific literature. And so we quickly found common cause in collaborative research. The human neutrophil NADPH oxidase is critical to killing of microorganisms by neutrophils. It sits in the plasma membrane of the neutrophil, and then it takes receipt of reducing equivalents from cofactor NADPH, hydride transfer, which is then delivered as 2 single electron transports to molecular oxygen to produce superoxide O2-. And superoxide production is critical to microorganism killing by neutrophils. Later, the superoxide dismutes spontaneously to produce oxygen. Two superoxides go to produce one equivalent of oxygen and one equivalent of hydrogen peroxide. So the Verona team purified human NADPH oxidase to homogeneity, and one of the team brought the enzyme preparation over to Oxford for me to do studies. We incubated it with the DMPO and saw, as in the control, and saw no signal, monitored by electron spin resonance. We then added the cofactor NADPH, and we saw this lovely signal that represents formation of superoxide. I'll just put the pointer on here. This is a characteristic signal of the superoxide adduct. And when you add varying increasing amounts of NADPH, you get increasing signal. The adduct is not perfectly stable and degrades slowly over 30 minutes, so we see this slow decline. At that point, it wasn't known if NADPH oxidase produces only superoxide or maybe hydrogen peroxide as well. And one thing we deduced that if we add the spin trap and trap the superoxide, we prevent the oxygen reformation here. So that if we looked at the oxygen consumption by the enzyme in the presence of the spin trap, B here, if only superoxide is produced, then the rate of oxygen consumption doubles because no longer do we allow this reformation of oxygen. And that was indeed found. And so we've provided the first demonstration that this enzyme produces only superoxide. And Filippo is a lifelong friend, and he also the members of his team I've known for many years, and we collaborated later when I moved to start my own independent research group. And when I started my own independent research in 1984, I chose for subject of study glycation by the reactive metabolite methylglyoxal. And over many years, we've characterized the principal reactions. The principal reaction with proteins is reaction with arginine residues to produce an adduct called a hydroimidazolone. We called it MGH1, this major isomer. And it's one of a group of compounds called advanced glycation end products, but it's actually the major one in physiological systems. And methylglyoxal reacts with DNA. Also, it modifies deoxyguanosine selectively to make a tricyclic imidazopyrinone adduct, and this we called MGDG. The extent of modification is 1 to 5% of proteins and 1 in 10 to the 5 nucleotides in DNA, which is large for a spontaneous formed adduct. The consequence of this for proteins is that the proteins become unfolded and activate the unfolded protein response, and for DNA causes mutagenesis and in some cases carcinogenesis. The physiological defense is the glyoxylase system. The glyoxylase system is an enzymatic pathway present in all human cells, catalyzing the metabolism of methylglyoxal to delactate. It involves two enzymes, glyoxylase 1 and glyoxylase 2. Glyoxylase 1 catalyzes the detoxification of methylglyoxal to S-D-lactoglutathione, and glyoxylase 2 catalyzes the hydrolysis of S-D-lactoglutathione to delactate, reforming the glutathione consumed in the glyoxylase 1 catalyzed step. There are wide applications for glyoxylase studies in obesity, diabetes, and diabetic complications, which were my main area of study, but also cancer, chemotherapy, and multi-drug resistance, neurological disorders, aging, diet and health, including our later development of dietary bioactive GLO-1 inducer, and others. When I started studying this area, nobody was really sure even if methylglyoxal was produced in human metabolism, and nobody was sure what the glyoxylase system did, although they knew it was widespread. This was lesson 2, that pathways of human metabolism of unknown function offer an investigational challenge of potentially profound importance. And I hope in the subsequent slides you'll see that we have led to some findings that have profound consequences for human health and disease. The Inspirational Scientists, which really stimulated my pursuit of this research were 3 Nobel laureates that worked on dicarbonyl glycation, or at least methylglyoxal formation, and left research unfinished. And remarkably, I came in contact with all 3. Sir Hans Krebs discovered the citric acid cycle, which we also call the Krebs cycle, and he worked in the laboratory until his last years. And you may notice that coincided with the period of my PhD. And when I did the study on vitamin K-dependent protein synthesis in the Department of Clinical Biochemistry in Oxford, Sir Hans Krebs was the researcher just down the bench from me. And remarkably, he came to ask me questions about reactive oxygen species. I was astonished because I thought Sir Hans Krebs knew everything. So I was delighted to see that he would come to ask me questions. And I also thought at that time, being a young PhD student, professors knew everything. Of course, now I'm a professor myself. I know professors certainly do not know everything, and we're always learning. Secondly, when I started working on the glyoxylase system, the best known researcher was Professor Albert Szent-Györgyi, who won a Nobel Prize for discovery of vitamin C early on in his career in the 1930s. And I wrote a letter to him saying, I want to study the glyoxylase system, study its function and role in health and disease. Do you have any advice? And he sent me a very nice personal letter back saying, unfortunately, he'd forgotten everything he ever knew about glyoxylase. But for many years after, I framed that letter and had it hanging up in my office when I was an early stage career academic. And then finally, when I went to give a seminar at the Fox Chase Cancer Center in 1996 on glyoxylase, I got to have a meeting with Erwin Rose, who had been measuring methylglyoxal production from triphosphate, and he was later to share a Nobel Prize for protein ubiquitination in the proteasome proteolysis. And actually, we had 30 minutes of what I consider to be the most intensive debate and discussion about methylglyoxal formation and metabolism. And at the end of that brief talk, he said, "Paul, you are a good scientist." At the time, I thought it was just a very polite thing to say. Of course, later when he was awarded the Nobel Prize, I was, highly delighted to have a Nobel Prize winner to have said that to me at that time. So these 3 people really inspired my early stage research. I would have to say also I've been inspired by outstanding female scientists, one of which is Professor Eva Kona, who, as a medic, realized diabetic patients were developing visual impairment when nobody thought about diabetic, or was aware of diabetic retinopathy. She founded the field of diabetic retinopathy, and through her research, she has improved the lives of innumerable diabetic patients. And following her inspiration, I think that in the near future, I expect we will have an effective treatment for diabetic nephropathy. A further researcher I met was Professor Linda Partridge, FRS, who's a leader in biology and genetics of aging. Most of us will aspire in our career to have at least one paper in Nature or Science. Linda has 39 papers in Nature and 20 papers in Science, and she is by any measure an outstanding biomedical researcher and an inspiration to us all. And then finally, Professor Naila Rabbani, who works in my own field of dicarbonyl glycation. Naila came into research as a mature student and took on several very challenging and difficult research projects, working them through to success. She also, in the academic setting, took on the role of equality and diversity officer, ensuring that people of all backgrounds, all colors, all creeds and genders get fair representation and treatment within the university research setting. These are my inspirational researchers. Next, I would like to talk briefly about scientific literature and dedication, which is where I think the good ideas come from. Historians of science have studied scientific discovery and have identified common characteristics of the process of key scientific discoveries. And a common feature is investigators study the scientific literature. This is not just a bibliographic listing, but cross-checking and corroborating key findings, identifying well-evidenced experimental observations, inferences made from them, and identifying studies that are flawed in design, implementation, and interpretation, and of course, giving less weight to these. And this is lesson 3. To make next step advance, we need to know our area of study to the limit of current understanding and the robust evidence supporting it. Then, remarkably, historians find a period of reflection is important, where investigators consider new possibilities, likely interactions of metabolites and proteins and pathways not considered before. In fact, one of the well-known sayings of Professor Szent-Györgyi was, to be a scientist is to see what everybody else has seen, but to think what nobody else has thought. And this is, of course, the art of discovery. And then to develop new hypotheses and design experiments to test them. And if a hypothesis withstands all challenges we make to refute it, then it's a secure advance. Sometimes a hypothesis may seem secure, but we need to refine it further later. And this is lesson 4 that many historians of science will also support, that secure advances in science occur mostly in small steps, building on existing knowledge, advancing and refining previous work. So when we hear of Nobel Prize winners, it seems like a major rapid breakthrough, but actually Nobel Prize winners have spent many years in the laboratories and many hours thinking over their work and advancing their studies to the point where they're deserving of these prestigious awards. Early on in my research on glyoxylase, I developed and validated all analytical methodologies I needed to do my research, something I later called the Glyoxylase Researcher's Toolkit. And this is very important, actually, to make sure you're using very secure methods in measurements in your research. So I developed all the methods for measuring key metabolites, Mg, GSH, S-lactoglutathione, D-lactate. In some cases, I prepared high purity analytical standards where they're not available commercially, methods for analyzing the glycation adducts, MGH1, MGDG, methods for studying genetic polymorphism, expression, protein, and activity of the glyoxylase enzymes, later developing mathematical models of the glyoxylase pathway. And importantly, I shared these with colleagues at international symposia, and we developed best practice. This is lesson 5. Good research requires secure and validated methodologies. Perfect them and share them with others. You'll then make key discoveries, and you'll also become a sought-after collaborator. I also continued my professional skills development. Of course, originally I was a chemist, then for my PhD moved to biochemistry, and then developed advanced technical skills of stable isotopic dilution analysis, LCMS-MS, which is the leading technique for quantitation of metabolites in our field, used worldwide now, and omics technologies. I also developed mammalian cell culture skills, which I did myself and trained others to do so. I trained and developed skills in animal experimentation, and I trained in leadership, design, implementation, and data analysis and interpretation of clinical studies, of course, working with clinically qualified investigators, co-investigators. And until recently, I worked alongside the researchers in the laboratory. This is lesson 6, to make advances in multidisciplinary research, become a multi-skilled investigator, and often you'll find yourself leading a multi-skilled team. One of the first watershed outcomes of this research was the first report of increased methylglyoxal in blood samples of clinical diabetic patients, where in this ribbon graph you'll see methylglyoxal concentrations in blood are skewed over to high concentration ranges in diabetic patients, increasing 5 to 6-fold in patients with type 1 diabetes and 3 to 4-fold in patients with type 2 diabetes. I did this in collaboration with Professor Peter Sonksen, a leading diabetologist at St Thomas's Hospital at that period in London, UK. And Peter has remained a lifelong friend and currently a Facebook friend with me now until the present. Next, a brief comment on teamwork and collaboration. As you may well have realized by now, biomedical and translational research often requires a multi-skilled team, bringing together complementary expertise, working together on interdisciplinary problems. And this is often where key innovations are to be made, often collaborating and leading international teams. If I look at the team involved in our diet and health studies and GLO1 inducer development, you'll see that I had Professor Naila Rabbani with me, expert in proteomics and metabolomics, or ageomics, for our particular focused area. Martin Weikert, a clinical endocrinologist. David Rand, one of the leading mathematicians in the UK and internationally. Professor Till Brettschneider, a leader in quantitative digital image analysis. Tony Smigel, a leader in advanced microscopy. And Guy Barker, a plant genomics expert. And it's often said, but it's absolutely true in this case, I could have not have done this research without the experience and expertise of all these leaders, co-PIs working with me on this research. And that is lesson 7. The efforts of the team is much more than the sum of the parts. When you bring complementary skills together, you get incredible synergism for advance and innovation. And indeed, my team has collaborated with most of the leaders in diabetes research of the last 2 decades worldwide. Peter Nafrod, Christian Hansen, Peter Rossing in Europe, USA, Michael Brownlee, Andrzej Królowski, Paul Beiswinger, Australia, George Jones, Mark Cooper, and in Japan, Masi Yamamoto, working on the transcription factor NRF2. And of course, I'm very grateful for their collaboration and support. I also led research on glyoxylase in cancer. I developed the first cell-permeable glyoxylase 1 inhibitor, which we found had anti-tumor activity in vitro, and then collaborated with the leading experimental oncologist in UK at the time, Professor John Double, to find anti-cancer activity in tumor-bearing mice. And then going on, we went on to work with Professor Mike Stratton, the director of the Wellcome Sanger Institute and leader of the Cancer Genome Project in the UK, and with his then PhD student, Tom Santarius, to discover GLO1 amplification, gene amplification in cancer. And later with Professor Martin Weikert, who's director of the Neuroendocrine Tumor Centre of Excellence in Coventry, UK, to discover the high prevalence of GLO1 amplification in neuroendocrine tumors and it's being a strong negative survival factor. Again, this is the strength of teamwork and collaboration to make advances in innovation and discovery. Then finally, I feel I have to address some contemporary concerns. Of course, I'm an old hand in biomedical research now, and I want to bring to your attention contemporary concerns and hopefully provide you some guidance to how we can work for improvements in biomedical research in the future. And these 3 concerns are the reproducibility crisis, predatory journals, and to reimagine research, a call by the UK Wellcome Trust. Firstly, reproducibility crisis. This initiated when a researcher, Glenn Begley, moved to the USA to take up a position as vice president in Amgen of hematology and oncology research. He reviewed 53 landmark breakthrough projects from leading laboratories and researchers. And after the publication in the subsequent years, he found 47 of the projects had been discontinued because the original findings could not be replicated. And then he developed this further and had a publication in Nature about the lack of reproducibility of many biochemical research studies. And this is where he coined the phrase reproducibility, or sometimes replication crisis. So is there a reproducibility crisis in biomedical research? Nature magazine followed this up itself and published a survey in 2016, surveying the views of 1,576 researchers, and not just in biomedical research, but across the disciplines. And surprisingly, about half researchers have not been able to repeat the work of other researchers, and surprisingly, they can't repeat some of their own research, which shows that there are some systemic issues with the way sometimes we do research that impairs a very important aspect of our research, that it should be reproducible. And Begley put together 6 rules of reproducibility when we assess a research publication. We should ask, were the studies blinded? And of course, for clinical studies, were the patients randomized to placebo and treatment arms? And for animal studies, were the animals in different study groups, their cages, were they randomized throughout the rack of cages to control for any effects on different location in a rack of cages? Were all results shown? Because sometimes researchers will take out some of the results for publishing and then sort of bias the report, of course. Were experiments repeated? Were positive and negative controls shown? Were reagents validated? As I previously said, one of the things I did myself early on in glyoxylase research was make sure I had high purity reagents for the analytical techniques. And were statistical tests appropriate for the study? So do we have a reproducibility crisis, or on the other hand, do we have an opportunity to improve in the future? I think the latter is the way to look at it. There are some, let's say, legitimate causes of irreproducibility, which nevertheless we need to address. Studies that are underpowered. This is common in exploratory clinical studies where it's been difficult for the investigator to attract sufficient funding to test the hypothesis. We need to work on that, to how to avoid that. And probably, I think, one of the main reason for sometimes limited reproducibility is unawareness of the investigator of interferences and confounders influencing experimental outcomes. Particularly, example would be poorly validated reagents or controls. If you're relying on commercial reagents that have of variable purity, for example. Unlegitimate causes of irreproducibility are lapsed integrity by investigators, which are more commonly referred to as, of course, fraud and bias, fabrication of data, manipulation of images, and selective reporting of data. Actually, for fabrication of data, it's thought that only 1 to 2% of researchers are involved in this, thankfully. Manipulation of images, I think, became more common when with digital photography and also programs to manipulate images post-capture. And we have to address this, and measures to address this have been taken by many journals now. So we need to prevent this. We need to put in prevention and controls to improve reproducibility. And many research institutes, like the research institutes in the Qatar Foundation, have done so. We need to improve governance in the research laboratory. Journals need to be more supportive for publishing replication studies, reproducing breakthrough studies. So we can demonstrate replication or not. And we need to improve surveillance of experimental evidence during the peer review process and post-publication. And we all should be mindful, of course, that there are centers for scientific integrity throughout the world that are doing surveillance of post-publication images to look for evidence of image manipulation. And secondly, predatory journals. And this is an issue that emerged in the scientific community probably 5 or 10 years ago, and the scientific community was slow to respond, mainly because it's been difficult until recently to get a consensus definition. A definition of a predatory journal are entities that prioritize self-interest at the expense of scholarship and are characterized by false or misleading information, deviating from best editorial and publication practices, with a lack of transparency and or aggressive and indiscriminate solicitation practices. And And there's been a survey of journals across the disciplines shown on the right-hand side at the top there. And there are over about 11,000 suspected predatory journals. And thankfully, there are over 22,000 legitimate journals. So when we consider this, where should we publish? So if we look at the fundamental principle of why we publish to make other investigators aware of our hypotheses and advances and to share sufficient detail of the supporting experiments to enable repetition. Replication is a key factor again coming up here. Publishing in legitimate journals provides the reader some assurance of the quality of the work. It has passed editorial scrutiny and thorough peer review. So like me, I'm sure you aspire to publish in leading journals of your field of expertise, like Nature Medicine, Diabetes. My personal view is try to publish in impact factor journal for greater than 4. I say this because this is typical of the impact factor of journals affiliated with leading scholarly and professional societies. There are some exceptions. Always aim for a better journal in the future. And also consider doing an occasional authoritative review and opinion piece, because it shows your peer group that you're really up to the leading edge of understanding in your field. But restrict yourself to your field of expertise, otherwise your review will not be seen as authoritative. And this is lesson 8, only publish in legitimate, reputable journals. The final contemporary concern is to reimagine research. And this arose from last year when Wellcome Trust asked over 4,000 researchers in the UK and internationally their views of the research working environment. The outcome was disturbing. Only 33% thought the working environment was positive. 55% thought it was negative. When asked for some descriptor words, there are some positive words there: collaborative, competitive in the sense that we hope our work is competitive with the best done by our peers, and supportive. But there are also some negative terms there: pressured, stressful, toxic, unsupportive. And this is really, really disturbing for old hands in the biomedical research community, like myself. And the Wellcome Trust then said, if we aren't working in a culture that's creative, inclusive, and honest, then we're not getting the best research. And of course, in QBRI, HBKU, and QF, we want the best research. So we want a culture that is creative, inclusive, honest, supportive, rewarding, inspiring, and stressful, and not stressful, not stressful, and successful. I mean, sorry. And when I considered the campaign in the Qatar Foundation of I Am QF earlier this year and the QF culture values, I see that they're much in keeping with the Wellcome Trust reimagine research of explore fearlessly, empower others, exceed yourself, and extend compassion, and the behaviors of accountability, collaboration, transparency, and innovation. And this is lesson 9, that the QF values and behaviors will help us improve the research culture and help us work with the Wellcome Trust to reimagine research and make the research environment a much happier place for people to work in. So finally, I just consider briefly where we stand now, 36 years on from when I first started glyoxylase research. Well, obesity and diabetes and diabetic complications. We know methylglyoxal is a key role in insulin resistance, type 2 diabetes and diabetic complications. We know glyoxylase 1 is a pro-longevity factor from studies and healthy aging from studies in the nematode C. elegans. We know glyoxylase has a role to play in cancer, in pre-malignancy. It's a tumor suppressor. But when we've developed a malignancy, glyoxylase 1 switches from protecting the human host to protecting also the tumor, and then it becomes a negative survival factor for patients and also a factor linked to multi-drug resistance. For therapeutics, we have GLO1 inducer dietary supplement for the prevention of type 2 diabetes. We have GLO1 inhibitor for the treatment of high GLO1 expressing multi-drug resistant cancers. And we've been working recently on GLO1 related therapeutics and prophylactics that are in development for COVID-19. In pursuit of knowledge, you can see over the years my appearance has changed quite a lot, from a youthful young PhD student in 1980 to now 2020, a somewhat more older looking person. In my career, there have been challenges, of course, the relentless task of seeking funds for the research team, reviewers of funding applications and manuscripts failing to appreciate the full merit of the research proposal and research outcomes, respectively. But there have been many more delights and fulfillments, particularly pursuit of knowledge, discoveries, and understanding of how and sometimes why cell and human physiology works the way it does. Training, guiding, and working with and inspiring others in the pursuit of knowledge. Friendships, fellowship, and appreciating abilities of colleagues worldwide. And lesson 10 for me is overall, I've had a very good time in biomedical research. I hope to have some more very good times, and I very much hope you will do too. So in the final credit slide, I'd just like to thank my current team, Dr. Ming, Dr. Alberto, Dr. Patrick, working with me in QBRI, and the PhD students, Mariam and Aldana, all the collaborators in QBRI elsewhere in Qatar, and collaborators beyond. And of course, I must thank most greatly and graciously the Qatar Foundation and Her Royal Highness Sheikh Mosa for the generous support of my research in QBRI. And I also thank you for your attention. Thank you.
Thank you very much, Dr. Paul. And thank you for sharing your inspiring journey in research and for sharing all your insights and lessons with us. Before moving to our next part in the webinar, I would like to remind our audience to please send their questions using all panelist option in the Q&A tab. So I would like to ask you a couple of questions before moving to the audience questions. The first one, in your experience, does institutional prestige affect young researchers' career prospects, especially when sending manuscripts to publisher or applying for funding and grants?
I think the originality and the strength of the science is the major consideration, but of course, support of your institution for your research application can have, and the environment can have a role to play. So it's very important for the host research institute or department to be supportive of their young career investigators when they apply for funding applications. They need to show that if the application is successful and funded, that the environment will be well supported by the host institution. I've sat on grant review committees in the UK and internationally, and I know, and I would like to reassure everybody that there is no prejudice towards institutions. The main priority is the strength of the science and the hypothesis to be tested. But nevertheless, it is very important, and I encourage all executive directors, all department heads to support their young researchers strongly when they put in funding applications.
Thank you very much, Dr. Richard. Dr. Paul, sorry. My second concern for students and young scientists facing the transition from their phase of receiving education to the self-responsibility of creating a career and a professional life. What would be your advice to them?
I think as generally is the career progression to develop your skills as a PhD student and a postdoctoral researcher. If the opportunities arise for funded, well-funded postdoctoral fellowships, like the Wellcome Trust, for example, runs some very good fellowship schemes, and many other funding organizations. That provides you a well-funded fellowship, provides you a sort of interim step between having funds to do your own research, a small research team, to establishing then fully your own independent research team, where you have to, as I mentioned, you have to keep applying for funding, external funding usually, to keep you to support your research team. So it's a process of really developing your skills, developing your professional network, looking for good potential funding sources of funding and environments to base yourself, research institutions, well-founded research institutions. And then, inshallah, you'll be okay.
Thank you very much, Dr. Paul. So moving now to the last part of our webinar. Yeah, let's go over the audience questions. First one. Unfortunately, the publish or perish phenomenon has resulted in many researchers cutting corners and applying some of the fraud practices you described in your presentation. Can you suggest some ways as to how young researchers can cope with this pressure?
I think young researchers, of course, need to stand firm and do good work and make sure they get sufficient support from their institute and their team leaders to repeat experiments sufficiently until they are sure that they've got secure results. I think for some time now, it's the publish or perish phenomenon has been receding somewhat in well-found and good quality institutes and departments. It's not the number of papers that you publish, but it's the quality of the papers you publish and the contributions they make and how often they're cited when they're published. So I think this phenomenon is retreating. I think there's respect for very good, solid research that is reproducible, can attract a lot of citations, and if you can hold firm with that, there are many old hands like myself that were definitely around to support you if you come under any adverse pressure in this regard, and you'll be well respected in your field. By developing robust analytical methods in glyoxylase research, I became a sought-after collaborator internationally. Many of the collaborations that were established in leading diabetes researchers came to me because they knew that I'd developed robust procedures in my research, and also I'd gained a deep and thorough understanding of the research topic as well. And this is what I would recommend young researchers to do as well.
Thank you, Dr.
I hope that
Paul.
answers the question.
Yes, perfect. Is there a governing body to audit top journals for fairness, transparency, and fixing the impact factor? If not, should be there a governing body how WISH can highlight this issue?
Yeah. Putting this presentation together, I was thinking that the reproducibility issue is something that WISH, one of the QF organizations, could address, maybe in association also with the Wellcome Trust concern for re-imaging research. I think that would be a great thing to pursue and to explore. There are different organizations in the USA. I think there's a organization for integrity in science, and they have people, as I said, doing surveillance of published images, looking for manipulation of images, and they will bring any manipulation of images they find to the attention of journals. I think the journals themselves are taking more action now. One journal, Biological Sciences journal, that I get the weekly content of the journal in my email account, I see regularly the journal is withdrawing or papers where they've found problems. So there's a lot more surveillance by journals. And there are various websites, like there's a website called Retraction Watch, which keeps an eye on studies that are retracted, or many researchers have called for. Researchers can write to journals and give an expression of concern if they've not been able to reproduce studies. And so this is something also coming now into our profession. And I think this will help. The best way to do this, of course, is prevention. And all these surveillance measures will help. I hope that helps. The answer helps too.
Yes, perfect. Thank you, Dr. Paul. You work closely to Nobel laureates. How does a young researcher overcome the imposter syndrome with confidence when working with experts?
I think certainly as a young PhD student, I felt in awe of Hans Krebs. I wouldn't dare go up to ask him a question. And I was a bit overwhelmed, frankly, when he came to ask me a question, because I thought, I was very early on in my career. So you'll find that the people that have made great achievements in biomedical research often turn out to be some of the most humble, and will be quite easy to approach and discuss issues with, and often are the most free of giving of their time as well. And of course, they have great experience. So I think take confidence that high achievers in biomedical research are only too happy to engage with young researchers, give them benefit of their experience and guidance wherever requested.
Next question. Thank you, Dr. Paul. According to you, is it more rewarding to be in academia or pharmaceutical industry?
I think in academia, you get more freedom to pursue a research argument of particular interest. It would have been very difficult for me to do glyoxylase research in the pharmaceutical industry, because when I started out, particularly, nobody in pharmaceutical industry was interested in that. So you have more freedom to explore research argument. In the pharmaceutical industry, of course, you have your resources provided for you. You don't have to spend time writing research grants, funding applications, although you do have to make a case to your inline manager why you're deserving of the funding to do work. And of course, the strategy is very much set by the senior management, quite detailed. I think it can be rewarding in both sectors, and there are some leading experts in diabetes and obesity research that have spent times in industry, in academia, gone to industry, and then come back to academia again. So, and it's very nice to work with a pharmaceutical industry and the food industry, as I've done. You can learn things from them. Their close management, detailed management of research projects, for example, which auditors would be very much encouraging of, to find that in industry they're very, very good on management, detailed management and reporting of projects and progress. So I think it can be rewarding in both sectors, and sometimes you can move between both. But if you have a particular research area you want to pursue, and it's not active in the pharmaceutical industry, then it's very difficult, of course, to go there. You have to pursue it in academia, really.
Yes, absolutely. Thank you. What can be changed here to be able to make QBRI as good training center for young researchers, so they can make a good careers in science?
Yeah, QBRI is a very good center, of course, for researchers. We're very well supported by the Qatar Foundation. I think if we reflect on the IAM-QF behaviors and values, as I mentioned, and also the conclusions of the Wellcome Trust recently, then, and we implement the wishes of the Qatar Foundation for our working environment, and which is supported by the Wellcome Trust, then we can make what is a good environment to work in QBRI, we can make it even better. And I hope we can do that we can make the working environment hit all the positive words in that word cloud that I mentioned, and none of the negative words, because I hope I've got it across to you, but a career in biomedical research can be very fulfilling, very rewarding, and you can have a very good time in the process of research and discovery and innovation.
Next question. The current research focus in Qatar now is moving towards generating funds by providing services to researchers within Qatar and internationally. This becomes a bit monotonous. What are your thoughts on this?
I've done some service analysis myself in my research career. When we were in the UK, we did some service analysis for Nestle and for Merck Pharma in Germany. So it can be quite rewarding and financially advantageous for your institute if you do this. Often you find that you need to have a unique selling point. You're doing some analysis that is not routinely available commercially, because if you try to compete with commercial analysis labs, then that's what they do all the time, and it will be difficult to compete. But if we have a USP for providing an analytical service, then it's a valuable thing to do and can be quite lucrative. It also provides for diversification of funding support, of course, which I think Qatar Foundation probably has in mind as well. That if we can make income from providing services, we're helping our clients, and we're also helping ourselves by income generation. Regarding a bit monotonous, I think if you're at the leading edge of analytical techniques and technology platforms, it's not monotonous. Actually, it's you're at leading edge, you're at the forefront of innovation, and maybe you get the chance to do some modifications to the platforms to even make them even better. Then you're in the process of discovery and innovation, of course.
Perfect. Thank you, Dr. Paul. Next question. I think we have time for a couple of questions.
Yeah.
Reproducing results is important, and more of these studies need to be published, but likewise, many negative results never see the light of day because those are of lesser interest to journals. How can we make these studies more visible, like journals that publish negative results, for instance?
Yeah, I think important negative results are good to see published, and particularly if in finding a negative outcome, you can build on that to refine the approach and make a new discovery. Now, I myself was in the glycation field, was involved where I couldn't repeat studies published in a paper in Nature. And I did eventually get the study published, but not in Nature, actually in Biochemical Pharmacology, and which has been very important. Nevertheless, it was very important to realize there were problems in this high impact factor study. So I think if you find an issue, a negative outcome, if you can then not only find that one avenue of discovery seems to be cut off, if you can then turn your focus and find a new one opening up, then that will help you attract a publication. I mean, for example, if we find that one bioactive is not an inducer of glyoxylase 1 that somebody has published, but we find another one that is a good one by looking at related compounds, then that helps. And it helps get the publication, and it helps move the field forward. What seems to be a negative aspect, nevertheless, to be turned into a positive.
Thank you, Dr. Paul. The last question. I always have the passion for the research, but I had to stop for 8 years due to motherhood. And now I found that I have a huge gap and lack of experience. What's your advice in such cases?
Well, that is, of course, I gave the example of Professor Naila Rabbani, who came into research as a mature student and has still been able to build up experience and expertise to become a leader in a research field. So I would say, of course, there's many cases where you have to take time out of a research career for family reasons, but nevertheless, you've got the skills, the expertise, the dedication and commitment, then certainly come back into research. And the gap in your career people should take allowance for that. Of course, you have to produce the achievements, but I wouldn't let that put you off. It's part of life, and people reviewing appointment applications for appointments and funding applications will take into account these normal life events. These are normal life events, and we're all respectful and sympathetic to those.
Thank you very much, Dr. Paul. With this, we conclude today's webinar. Thank you. I'm really personally feeling inspired and motivated after your talk. And thank you all for joining today's webinar. If you have further questions, please feel free to contact Dr. Paul or us through email. I'd like to extend my thanks to the fantastic team working behind the scenes, Dr. Adviti, HBKU and QBRI Communications, Reem, Omar, and Azzuddin. Just one final thing. When you exit the webinar, we would appreciate you filling out the survey that will pop up on your screen. Stay tuned for more inspiration in our next webinars. And thank you very much.